to welcome Dr. Razzaque. As you can see from his title slide, he is joining us from the U.S. And he's sort of got a dual personality, if you will, in that he's actually sort of existing both at the Lake Erie School of Osteopathic Medicine in Erie, which is a few hours away. But then at the same time, he's still in Cambridge and has some projects ongoing there at the dental school. And as you can see from his talk, his title, he's really interested in different kinds of hormonal factors that play a role in how the body handles levels of phosphate. And I think what he's wanting to do this afternoon is really just give us a sense, in very broad terms, some of what he's done over these last couple of years, looking at endocrine factors relating to how the body handles phosphate, and then what can happen if phosphate levels become dysregulated in terms of shifting risk profiles for various kinds of degenerative disease. So I think with those few words of introduction, I'll turn things right over to him and thank him for coming.
Thank you. Can you hear me there? All right, good. So this is really pleasure and privilege for me to be here, and thank you, John, for the invitation. I really appreciate it. When I sit in a biomedical science or clinical science presentations, it's mostly related to understanding a disease process or how to diagnose a disease early, how to provide better treatment to a patient, better care to a patient, better service to a patient. It's all about the patients. It's about them. Today, I will share with you some information that is important to us, you and me, who are blessed to be healthy. So I will show you endocrine regulation of phosphate homeostasis and explain why its dysregulation should be of concern to the healthy individuals like us. So I will show in first part physiologic regulation of phosphate homeostasis and tell you a little bit about the consequence of phosphate toxicity and end up my talk with some recommendations. So let me tell you why understanding the regulation of phosphate homeostasis is biologically important. It's an important component of nucleic acids. It's also an important component of cell membrane. Intracellular anions are involved in activations and inactivations of enzymes and coenzymes. Phosphate also plays a role in cell signaling, energy metabolism, and bone mineralization. So it doesn't matter what system you're working with. If you don't have optimum cellular phosphate balance, it's going to be affecting that cells and eventually that system. So it's very important to have optimum phosphate balance. In this slide, I listed some of the systems that are affected when serum phosphate level is low. As you can see, musculoskeletal system, cardiovascular system, pulmonary system, neurologic systems. Almost all the systems in the body are affected when serum phosphate level is low. There are also consequences of phosphate when it's high in our body, and one of the most common being vascular calcification. So as you can see, like it's not only biologically important, it also has clinical importance to have optimum phosphate balance in our body. So this is a traditional view of how phosphate is regulated in our body. When serum phosphate level is high, PTH increase the excretion of phosphate through the kidney, and vitamin D absorb less phosphate in the intestine to restore the phosphate balance. This traditional view is correct, but there are missing links that we are trying to do and connect through our research with our collaborators in different places. So in this slide, I listed 3 diseases. These are big names: XLH, X-linked hypophosphatemic rickets, ADHR, autosomal dominant hypophosphatemic rickets, and tumor-induced osteomalacia or oncogenic osteomalacia. So these three diseases, these patients, despite they have like different etiology and some are inherited, some are acquired, their body cannot keep the phosphate. So it's going out in the urine. So their serum phosphate level is extremely low, and that's affecting their bone. As you can see, these are 3 XLH affected sisters, and you see like they have like this bowing of their legs because their bones are not strong enough. This is a very unusual situation. You have a patient where calcium is normal, vitamin D is normal, PTH is normal, but phosphate is very low. And it turns out FGF23, which stands for fibroblast growth factor 23, is very high in these patients. And this factor is inducing phosphate wasting from their body, and that results in serum phosphate level is very low. Okay? And I'll talk a lot about this FGF23 in my talk today. Okay? So what is FGF23? It's a 30 kilodalton secreted protein. FGF23 is mostly produced by the bone cells. FGF23 can suppress vitamin D activities. And also in the human, we have patients who are having FGF23 mutations, and that can show abnormal regulation of phosphate in their body. And this is the structure of FGF23. It has a FGF receptor binding domain. Also, there is a, I don't know how to bring this arrow here, processing site which makes it 2 smaller fragments. So this full length is the protein that is functional. What is the role of these smaller fragments is not known. Okay. How does FGF23 regulate phosphate balance? And this depends upon like how do we approach it. Different lab is approaching different ways. We, in collaboration with our collaborators, we try to do it through the mouse genetic approach. We generated a FGF23 knockout mice and asked 3 very specific questions. Where does FGF23 express? What are the functions of FGF23? And how does it exert its function? And one of the ways is done, a mouse FGF23 having 3 exons. So this was replaced with the Razzaque so that you can track that expression. And that is shown here. This is embryonic 12.5. You can see in the liver and maybe around the heart area there is expression. These are adult mice, 6 weeks. These are control, and these are knockout through the expression of Razzaque. You can see these bones are blue, which means bones are FGF23 producing cells. We wanted to know exactly what cells are producing this. So we took these bones and make the sections out of this, which is shown here. And all these blue staining cells, I'm sorry, this pointer is not working. That's why I have to move.
It's perfect.
These blue staining cells are FGF23 producing cells in the bone, which are osteocytes. So osteocytes are main FGF23 producing cells. There are also other evidences. This is in situ hybridization. Again, you can see some of these osteocytes are strongly expressing FGF23. But these are mice. What about the human? These are human bone. This is in situ. Again, you can see this arrowhead, these bone cells, which seems to be osteocytes, are expressing FGF23. This is a protein expression by immunohistochemistry. This CKD stands for chronic kidney disease. In the chronic kidney disease, their expression is high. And you can see these expressing cells in the bone are mostly osteocytes. So osteocytes are one of the major FGF23 producing cells, both in the experimental animals and in human. So now that we know like what cells is producing FGF23, we wanted to know what is the effect of knocking out FGF23 from the mice. And this is the result here. These are the littermates from the same parents. This is a wild type, and this is a mice that doesn't contain FGF23. So from the visuals, you can see these are smaller in size. Also, these mice, when you see their survival, they don't live too long. By 12 to 15 weeks, usually all the mice are dead. And this is their serum phosphate level. So these mice, when you take the blood, compared to the wild type, you can see these red bars. Their phosphate level is extremely high. So when you taking out the FGF23 function from the mice, they have extremely high phosphate level in the serum. And this, I don't have the time to show today. There are also transgenic mice where people express FGF23 very high and show there is opposite effect. Compared to the control mice or wild type mice, you can show their serum phosphate level is low. So it seems to be related to the systemic phosphate homeostasis. So what happened to their bone? Again, this is the wild type, and this is the FGF23 knockout mice. And you can see this is bone-cosa staining, and there are more minerals here. So seems to be in the bone there are more minerals. But you know, the bone is a very delicate structure. So it's depends where you're looking at. You know, some places you see there are more minerals, some places you see it's not exactly what this picture is showing. So we also look at like, okay, so we have a mice where like phosphate level is extremely high. So what is the effect of this high phosphate in the soft tissues? We look at the kidney. This is a wild type kidney, and this is a kidney that's taken out from the FGF knockout mice. So you can see the size is smaller. We make the section out of this kidney, and this is the wild type kidney. You can see nice structures of glomeruli, tubules, and blood vessels here. When you see the knockout mice, where phosphate level I showed is very high, you can see there is calcification. This is a blood vessel. You can see vascular calcification here, right? So not only like when your phosphate level is extremely high, you see the changes in the bone, you can also see it's affecting the soft tissues. And this is also present in the lung and other places. So when you do the mouse genetic studies, I'm sure John is doing it a lot. And when we have this distinct phenotype, like visually there is changes, there's biochemical changes, there is histological changes. These are very good. It's a good recipe for writing a paper. But we are not doing the research just to see all this phenotype in the mice. We have a bigger goal, like how does it connect to certain diseases? If there is a connection, then there is importance of doing studies with these models and try to understand certain events in those diseases. So we have a knockout mice. We take out the FGF23. We showed their serum phosphate level is high. We also found they have ectopic calcification, and all these things are happening because they don't have FGF function in their body because we are knocking it out. Is there any human disease that mimic these kind of changes? If it's, then it's make more sense to use these mice and try to understand certain events. So we find there is a disease that called familial tumoral calcinosis. Okay, this is an autosomal recessive disease. This patient, they have loss of function mutation in FGF23 that leads to serum phosphate level is high and also ectopic calcification in different places that is shown here. So now we have a human disease where human FGF23 is mutated and that causing the loss of function and leading to the features or phenotype that we find in our mice. So that was very interesting to know and encouraging us to do a little bit more research on using these mice. So I showed you when you are taking out the FGF23, serum phosphate level is extremely high. And it's not only in the mice, also you can find there are human diseases, there are similar changes. So how does FGF23 regulate phosphate homeostasis? Most of the phosphate absorption and reabsorptions are happening in the proximal tubules. Okay? In the proximal tubular epithelial cells, there are transporters that call sodium phosphate cotransporters. There are different types. There are 2A, 2C. So what happens when these epithelial cells are having these sodium phosphate cotransporters? They can, when this urine is coming, they can reabsorb that phosphate. Okay? So if you don't have it, it cannot reabsorb and it go out of the body through the urine. This is if you visited Brussels, you might have seen this. So how much phosphate would be in your body depends upon what is the activity of the NAPI in the kidney. If you have lots of activities of NAPI in the kidney, what happens? This phosphate that is coming to the kidney will be reabsorbed and going back to the system. If NAPI is not working, then there is no reabsorption and that phosphate will come out through the urine. Okay? So we went back and we said like, okay, we have the FGF knockout mice and we have shown like their phosphate level is extremely high. So what happened to the NAPI? This is NAPI2A. This is a control mice or wild type mice, and this is a knockout mice where I have shown like their phosphate level is extremely high. And this is NAPI, again, this is sodium phosphate cotransporters. You can see in the proximal tubular epithelial cells, their expression is very high. So if you look at this image, and we also did Western, and you can see they're also high. So one of the things that you can speculate looking at this slide is like in FGF23 knockout mice, NAPI expression is very high. So all the phosphate that is coming to the kidney is reabsorbed and it's going back to the circulation. That's why their serum phosphate level is high. But you know, in the science, with the speculations, you cannot go too far. You always have to prove, right? Especially, you know, when you submit the papers. So we wanted to know, okay, very good to see like this transporters is expressing high here. Is it biologically active? So one of the approach that we took, we said like, okay, in the FGF23 knockout mice, NAPI is very high. So how about we generate a FGF23 knockout mice where there is no NAPI? So we made with the NAPI knockout mice and generated double knockout mice. So we have a FGF knockout mice where I showed they have high phosphate. And then we have FGF knockout mice where there is no transporter in the kidney. This is a global knockout actually. And we went back and looked at their serum phosphate level. So again, this is wild type level and this is FGF23 knockout mice. You can see their serum phosphate level is high. I have shown you early. And this is also FGF23 knockout mice without the sodium phosphate cotransporters. And you can see their serum phosphate level is going down here. So where this phosphate is going, if you look at the urine, you can see when their serum phosphate is high, their urine excretion is low. When you're taking out that transporters, now you don't have any reabsorption, reuptake is going on. So all this phosphate is going out in the urine, and that reflects the low serum phosphate level in these mice. So now we can say like not only this NAPI expression is high in these mice, these are also functionally active. And that's what I summarize here. FGF23 mediated renal regulation of phosphate homeostasis is a sodium phosphate cotransporter mediated process. Again, you can sit there and say like, Mohammed, it's good. In the mouse system, you know, you're showing us a lot of impressive data, but what is the relevance with the human disease? So I picked up 2 papers that published earlier. These are human diseases where they find this NAPI2A mutation in the human causing renal Fanconi syndrome, which is like they are also wasting a lot of phosphate out of their body. This is SLC34A, which is synonym for the same molecule, NAPI2A. Again, they are showing in different papers in the human also. When you have mutation, you have problem with this that also related to dysregulation of the phosphate. So this is not only something that's happening in the mouse system animal experiment. This is also very relevant to the human. So how does FGF23 exert its bioactivities at the cellular level? For the interest of time, I will show you the summary slide here. Most of the FGF family, they need to bind with the FGF receptor to induce downstream signaling. For FGF23, it's very unique. Even if you have the receptor, it can be functional unless there is a cofactor which is called clotho is there. So you need 3 things. You need FGF23, you need its receptor, and you need a cofactor which is called clotho. When these 3 things are together, then it's become bioactive. We did a very simple study. We took human proximal tubular epithelial cells. These are cell culture studies. And you treat them with a vehicle or only with the FGF23 or the clotho. And we look for these phospho-signaling proteins. And you can see like when we put both FGF23 and clotho together, you can see these phosphoproteins are showing up, which means, you know, these are only bioactive when these 2 molecules are together. So I start my talk showing you this slide. What is the traditional view of phosphate regulation in our body, which is partly mediated by PTH and vitamin D, and that's why before people used to thought this is secondary to calcium regulation. With the identification of FGF23, this looks more like this with FGF23 that's coming from the bone, works on the kidneys, suppress the activities of the transporters to increase phosphate excretion. I don't have the time to show that side. FGF23 can also suppress vitamin D activities, and that can lead to less phosphate absorption and leading to low phosphate level. Okay. This is one of the reviews that I wrote a couple of years back, but most of the information here is still current. If you're interested to know more about that, I recommend you to read this. This is a bridge I drove, which is connecting these 2 great countries. And one of the reasons I'm here is to make some bonding, to set up some collaborations. And I'm very excited to talk to this morning with John, and hopefully we will be able to set up some good collaboration that will lead to some meaningful information. So I'll show you some evidences about phosphate toxicity. Okay, I showed you we have a mouse where there are a lot of phosphate. Okay, so what this high phosphate is doing? Again, this is a mouse work. This upper panel is from a mouse that is having normal level of phosphate, and this lower panel is from the mice that are having high levels of phosphate. Okay, and these are skin tissue. In the normal skin, you have this subcutaneous fat. Okay, And that actually keeps our skin very soft and smooth. In the high phosphate group of mice, you can see that subcutaneous fat layer is disappearing, okay? And their skin is very irregular and atrophic. This is BRDU, which is a marker of the cell proliferation. In the normal skin, you can see these cells are nicely proliferating cells. In the phosphate toxic mouse, you can see there are less proliferating cells. And tunnel staining is done to identify the number of cell death. Again, in the normal mice with the normal skin, there are very few this dead cell. But in the toxic mice, you can see there are a lot of these tunnel positive cells, suggesting these cells are dying. So there are less proliferation and more apoptosis or cell death that eventually lead to this kind of atrophy in their skin. This is, again, this upper panel is taken from the mice that are having normal level of phosphate. And this lower panel is from high phosphate. And this is intestine. And what you see here, there is distension, and these are the epithelium in the normal gut. And you can see there is obvious atrophy here, okay? And again, this is smooth muscle action, which I used here for identifying the smooth muscle layer here and also the blood vessels. And you can see, like, if you visually compare, there are a lot less blood vessel here. Okay? And all these things leading to atrophy. And same goes with the BRDU. You can see a nice BRDU positive nuclei here in the normal intestine, which is much less in the atrophic intestine. And all these things are caused by high phosphate in their body. And when you have this high level of phosphate, that induces infertility. So one of the interesting thing is, like, in these mice, when you reduce this phosphate burden, they become fertile again. So it seems to be like this high levels of phosphate is something to do with their fertility. Okay. This is one of the areas Ron is trying to work, phosphate toxicity and tumorigenesis. And this is one of the studies done a couple of years back. What they did, they took a mouse. I think, yeah, in their studies, they took the mouse that are mutated with KRAS. And those KRAS mutated mouse are developing lung cancer spontaneously. So they took that mouse and put in the normal phosphate diet and high phosphate diet. Okay? And you can see in the normal phosphate diet, there are some tumor foci in the lung. When you're putting them with the high phosphate diet, there are obvious increase in their tumor size and also tumor growth. So if you take a mouse that is spontaneously developing tumor, and if you give them high phosphate diet, put them in high phosphate diet, it seems their tumorigenesis is increased. Again, this is mouse study, and we have to see what is the human relevance of this kind of information. So reducing phosphate burden, either by genetic manipulation or by reducing the dietary uptake, you can suppress premature aging like features, at least in the mice system. So you can see, like, when there is phosphate level is extremely high, there are a lot of different things happening in the mice. What is the net effect of phosphate toxicity on survival? And this is a slide. This is the same mice line. Okay? We put one with the high phosphate diet, and we put another one with the normal phosphate diet. It's the same kind of genetic background mice. One is taking normal phosphate and the other group is taking high phosphate. And when you look for the survival, you can see the group that are taking high phosphate usually die by 12 to 14 weeks. Same group of mice, when they're taking normal phosphate, they have much better survival. Okay? So not only they have a lot of issues with different tissues that also reflect in their longevity. Again, these are mouse studies. What is the human relevance or clinical relevance of this phosphate toxicity? This is one of the studies that I picked up. These are chronic kidney disease patients, which means their kidney is not functioning. So their phosphate level is high in the body, okay? And these are the phosphate level from low to high, okay? And you can see this arrow, which is reflecting the relative risk of mortality and clearly showing when your phosphate level is high. And these are chronic kidney disease patients. So even in the human, you can see there are similar kind of trend with the high phosphate. But I'm sure you're sitting there and saying like, Mohammed, but we are normal people. These are about the patients. What about us? So what about us? I'm sure like a lot of you will be asking like, okay, if phosphate is doing that much of harm, how much is too much? Unfortunately, we don't know yet. We have to do more research. I'll show you an example that is published a couple of years back. So these investigators, they took heart disease patient, okay, and grouped those patients from low phosphate to high phosphate, okay. So these are patients with having heart problem, and then they group it from the low phosphate to the high phosphate, and this is the result look like. Again, this is the low to high, and you can see the hazard ratio of death in these heart disease patients are higher with high levels of phosphate. Okay? Now, if you look at this bar, where serum phosphate level is 3.5 to 3.9, and these are a pretty good number. N is like almost 1,000. Okay? And you can see like even this range, it seems to be like the hazard ratios is going up. I want to remind you the normal level of phosphate. The normal range of serum phosphate is 2.5 to 4.5. So I don't know how much phosphate is too much, but in certain diseases, in the kidney disease or in the heart disease, even the upper limit of normal range of phosphate could be harmful. And this is a study published a couple of years back, almost done on 10,000 individuals, and they excluded pregnant women. They also excluded diabetes, cancer, kidney disease, heart disease. So all these diseases which might be linked to the phosphate. So these individuals are kind of like you and me, normal individuals. And what they showed with the high dietary phosphate intake, that might be related to all-cause mortality. Okay? I think in this paper they showed like when it's go beyond 1400, it's become harmful. So I will show you some sources of artificially added phosphate food. These are the foods and drinks that are rich in phosphate, or the preservative that's are put in here. All these burgers, hot dog, pizza, all this soda that we are taking, these are containing high levels of phosphate. And here they are using phosphate as a preservative. Okay? So everything that preservative that you are buying and eating, most likely you are also consuming phosphate more than that you need to take in a day. So I listed 3 commonly consumed drinks here. And this is phosphate level. I labeled it here. So if you go to the market, you can have this calorie-free or sugar-free, caffeine-free, lot of different drinks we can buy, right? I want to remind you, none of these drinks are phosphate-free. So next time when you're taking it, remind yourself. So we live in a place where, like, when you put something, you have to put it in the ingredient level, right? So we look at it and we find, like, actually it's not like that the soda companies are hiding. They're saying it's containing, the sugar, caramels, and caffeine and all these things, but it also containing phosphoric acid, which is, so it's written there. The question is, like, do we know about it? So we did a very simple study, and there are studies that has shown, like, if you take this preservative-rich food, there are consequences. So anyway, we said, like, okay, there are foods and drinks that containing high levels of phosphate, and it's also written in the ingredient. Do we know about it? So we did a survey. We went to Japan in a medical and nursing school. We have 190 medical and nursing students who are around 21 years old, and asked them very simple question. Do you guys know the soda drinks containing high level of sugar? 99% say yes. I don't know why 1% doesn't know. We asked them, the same drinks is containing high levels of phosphate. Do you guys know that? Only 7% say yes. So you can see there is a huge awareness gap. Is it read? It's written there, but it's still, like, we don't know. Like, it's there. When we send it to the journal, they say, like, but you have to show, like, what this phosphate is, or this soda drinks is really doing. So that's something difficult. So we took 35 students. Again, these are around 21 years old. We tell them to come overnight fasted, and took the urine, okay, and give them a can of soda. On record, I don't want to say which one we give, but it's the commonly one that we consume. And after 2 hours, we took their urine again and measured the urinary calcium level. And you can see, just taking 1 can of soda, they are excreting more calcium out of their body. And these are healthy 21 years old boys and girls. So think, like, if this is happening to the elderly individuals who are having osteoporosis, taking vitamin D supplement, calcium supplements, and also drinking these drinks. So there are a lot of consequences of these drinks. This is another human study that we published. Especially for the kids, we say don't take too much sugary stuff because it will induce dental decay, or it will induce cavity in your teeth. Most of the kids are not liking that advice, but there are some kids actually listening to their parents, and still you see they have those cavities. So what else they can do? So we did a study, almost like more than 8,000 kids, around 10 years. So we found a group, they are taking low sugar and low phosphate, okay? And show, like, what is the dental decay. And then we find there is a group, they are taking high sugar and high phosphate. Of course, their dental decay will be more, right? And then we find a group, they are taking low sugar but high phosphate. So that's this group, and you can see like compared to low sugar and low phosphate, even though they are taking low sugar, by taking high phosphate, they are having more dental decay or cavities. So you can so there are independent effect of phosphate that can be seen in the human in different diseases. Again, like this is a very evolving area. You know, you have to do different areas to see what it's really doing, but it's causing a lot of harm in the body. So if phosphate is doing so many things, why do we miss it? What's the problem? Because it's not a rocket science, right? Because every time you go to the clinician, you always see like they're giving you the calcium, phosphate, and all these things are routinely measured. So why we are missing it? So again, this is an animal experiment. These are the rats. You know, you give them high phosphate from low to high, okay, from 0.5 to 2.5, okay, and then you take their urine and you can show like in the urine there are more phosphate is coming out. So you're taking more phosphate, your urine is dumping more phosphate. In the same animal, when you look their serum level, you don't see too much changes. Stays normal, okay? Despite their serum levels of phosphate is normal, when you take their body weight, you see it started to going down. What does that tell? Just because you go and you find your phosphate level is normal range doesn't mean it's not doing harm, because it doesn't always reflect in the serum level. In the chronic kidney disease patient, also like, you know, these things are coming in the very late stages or intermediate stage. And that might be one of the reasons, like despite we are having this calcium and phosphate is measured very routinely, still, you know, we are missing the harmful consequence of taking too much phosphate. So, phosphate toxicity can induce a lot of harmful effects in the body. It can impair bone mineralization, it can increase cell death, impair cell signaling, impair fertility, can induce vascular calcification, renal dysfunction, can induce tumorigenesis, and premature aging. This is one of the studies that Mutsuko, she is from Japan, she did this work in my lab and published this one. If you wanted to know a little bit more about this, I'll recommend you to read this paper. And when we published this paper, we have this news media, social media that picked up and tried to make the awareness. And I was very happy to see like one of your local journal or newspaper picked up our work. So that's good. So I showed you the diet or the food that is containing high levels of phosphate. These are some of the fruits that are containing low levels of phosphate. So basically, most of the healthy foods are good. You know. So I will end my presentation with a question that was asked a few years back in a cover page of a Life magazine, asking, can we stop aging? And this is what I think. No, we can't stop it. But with our efforts, we might able to delay it. And my recommendation would be avoid phosphate toxicity and enjoy a healthy life. Thank you, and I'll be happy to take any questions.
Well, I'll actually start with one question,
Sure.
since this is not really my area of relation, but the prevalence of some of the diseases that you mentioned in the beginning that are truly driven by the phosphate elevation, so the one with the bones, any stats on the prevalence of that?
people are doing on those areas. So I don't know, like some of these diseases are very rare. So it's not easy to find enough patient
Okay.
to come up with the real numbers. Yep.
So I'm sure it's complicated because, I mean, you pointed out lots of different areas of the phosphate influence. So if you capture all of those, you would say prevalence is pretty high. But I was wondering, yeah, for the ones that are maybe a bit rarer, where it's really the driver, what the stats were on that, and if it's also demographics, certain groups, certain areas of the world. Have you ever come across any information on that?
Not that I know. That I know. Yep. Yes, ma'am.
So,
I'm just using the mic so you can put it there.
so I was just curious about, you mentioned the phosphate influence on the intestine, and then you later on mentioned the decreased body weight that's associated with that. Is there a link that goes to the ability for the body to absorb nutrients because the intestine now seems to be damaged due to the phosphate? Is that a link that is kind of there?
That's definitely a possibility. Yeah.
So even something with, say, like celiac disease, because you mentioned a lot of the things that are high phosphorus containing, typically, I'm looking at a bunch of things on that slide that I know personally I can't eat, and I'm going, and I know there's a lot of unknowns associated with that disease as well. Is that maybe something that could be a link?
There could be some link. Yeah.
It's not my area either, but
Yeah.
Okay, cool.
Some of the things are directly mediated by the phosphate toxicity, and some are the secondary effect of that phosphate toxicity.
Right.
Yep.
Okay.
So it looks like your work has shown a pretty clear relationship between FGF23 and the sodium phosphate co-transporter. What exactly is happening, though, to connect those two? And there I'm wondering, sort of at the level of gene regulation, do you know, is this sort of an epigenetic mediated process? Are there changes in methylation, or is it something else?
People are working to find out that connection. It's not a random thing. It seems to be very well regulated. But what are the molecules exactly involved in that pathway? Those are not clearly defined yet. Yep.
Question. So, what's the consequence of removing the phosphate from some of these things that we shouldn't be eating? Like, would people notice if it was gone, as far as, I don't know, taste, or is there any push to get the companies to remove these high levels of phosphate?
Yeah, there are some issues here, like in the soda drinks, you put this phosphoric acid because of that tingering taste. So I guess like if you stop putting that one, it will be just a sugary drink. You will not take it. So, yeah.
Come through. So even then, that would apply to like the breads and the pizzas and the burgers as well. It's to maintain the flavor that's supposed to be
for the burgers and all these things, it's mostly put as a preservative.
So there would be alternatives then. Potentially, I don't know.
Potentially.
Yeah, you have to look at it. What happened, like when you're using the phosphate-based preservative, it's also retaining the water, so it keeps the thing moisture. So if you use like maybe other preservatives, when you take out from the freezer, it might be rock stone.
Russian zest.
Yeah.
Yes.
So there are some issues here, you know.
So with the feeding studies, and this is a pretty common question that comes up with any work of that sort, you were able to induce phosphate toxicity by supplementing the animal's diet. But how far outside of what would we say the usual physiological level did you need to go? So in other words, how high in phosphate were the animals' diets relative to what they would normally ingest?
So in our studies, the wild type mice or the regular mice are having like 7 to 8 milligram per DL levels of serum phosphate. And those phosphate toxic mice, they are having like 12 to 14, 15 milligram. So it's kind of like almost like double the level. But I don't think like you need to have that double the level to have this kind of toxic effects. Because in human, as I said, like when you test, you would hardly see any difference in the serum level until unless these are chronic kidney disease patients and all this, you know. And I believe like just because serum phosphate level is normal, it doesn't rule out like you don't have higher phosphate burden in the body. And that's one of the things we are missing. Yes, sir.
Yes, that was a great presentation, Mohammed. Just a quick question on kind of the regulatory side. I mean, I guess these additives are generally recognized as safe too, right? And I'm just wondering, you know, there's a Delaney clause in the U.S. Any kind of thing that causes cancer is kind of an issue with the food additive. I'm wondering, I mean, some of these additives, they're clean on chronic bioassays in terms of cancer development and stuff?
I think because these are in the market, so it's presumed like they are safe. That's why it's there. But you know, with the research and with the new findings, you started to see the things a little bit differently. And then you revisit and maybe you try to see like that range that is present maybe need to be revisited.
I'm wondering, has there been any activity, you know, in terms of promoting, you know, these studies to these agencies? I mean, because they look at stuff if they see it in the, you know, in the literature and in, I guess in the paper too, maybe not the Toronto Sun or something.
So one of the issues is like the phosphate is toxic. It's not very well known. So we are trying to show those things. And we have shown, and other groups also are showing it in the experimental models. And some people have started to pick it up in the human diseases. So I guess with more information, you know, it will have more impact on regulatory bodies to look at it and maybe revisit the whole things. But the whole thing is like phosphate is toxic is a very new area. It's an evolving area. One of the things that I try to do is go different places, show these images and try to show them like what could be the potential consequences that lead to two things. Generate a lot of interest to do more research, because we definitely need to do a lot of research to understand these underlying molecular mechanisms. And also, you know, there are studies hopefully in the human cohort to do more in-depth study to have a little bit better understanding of what this nutrition
I
things
guess
are
the
doing.
labeling is the same issue too. You see, you know, I can see why people, I mean, at least phosphate is on the bottle of Coke, but on a burger, you know, it's never on there. Right?
No, it's not in the, it's the requirement.
So there's no kind of, people are not gonna have any clue about some of these foods.
Right,
But,
right.
Yeah, and so I guess that goes along with the regulatory agencies for them to, you know, you're not gonna get food labeling changes until,
Mhm.
I mean, somebody gets concerned about it, right?
Right. I mean, I hope like they will start to appreciate like this is something that is important to be on the ingredient level, so people will know like what they are taking. But now that even like, you know, they know like phosphate is something bad, and we showed in our survey, they don't really know like where it's coming from because it's not labeled. Soda is a something like they're putting it. Again, that one of the things we hope like it will be happening in not very distant future.
Just takes a certain level of awareness, right? Like BPA in the models wasn't a big thing until
Yeah,
very late.
and you know, trans fat went quite quickly too.
Yeah,
Like,
that's another one.
I mean, from under the radar to
Yeah.
being an issue, right?
Is it listed on a nutrition, like I can't remember, but is it listed on the nutrition label, like your percent daily of phosphate intake? Like, you know how, because it has trans fat, it has sugars, it has iron, vitamin C, whatever. Is phosphate one of
No.
those listed?
No.
Yeah.
Because I guess it goes back to the question, how much is too much, right?
Mhm.
So what goes to, what should be ingested in at what quantity? What is safe?
Normally, like we should take 700 to 800 milligram of phosphate every day to have like optimum bone functions and other functions. It's not like we don't need phosphate. There are some studies I think there is recent US studies where they find like almost 35% people are taking more than the double the amount of phosphate they're supposed to take every single day. So just guess if they keep taking it for years and years and years, these will come with some cost, some consequences. But these are like long-term things. So hopefully there will be more interest in different places, and people started to appreciate the importance of looking at this. The very first slide I showed you, this is not something that is working in one single places. Your cellular function, from DNA to the cell membrane, to the signaling, to the energy production, everywhere you need this phosphate. So there should be very regulated and optimum amount in our body to have our cells functioning in a right way and correct way. So hopefully, yeah, I am hopeful. That keeps me motivated to do this kind of studies. A little bit out of box, but.
It'll be almost part of your last slide. That will be the driver, the aging aspect of it, rather than yeah. That's what people catch on. If you notice the articles, everyone caught on to the aging part, but not the other diseases.
Everybody's so scared of aging.
Exactly. You tell them that there's so much aging, then they'll be like, oh, forget it. Awesome. Thank you.
All right. Thank you very much.
Thank you.